Yeah, so my name's Leah Miller. I go to Dickinson College where I'm a math major and I'm from Connecticut.
Hi, my name is Saul Mota. I recently graduated from the California State University of San Bernardino, where I earned a Bachelor of Science in Applied Mathematics and a Statistics minor. I'm from Southern California.
Our research focused on Yersinia pestis, which is a plague bacteria, and it has 100% mortality without treatment, and there's currently no FDA-approved vaccine. So the Chopra lab, which is who we worked with throughout the project, developed two live-attenuated vaccines, LMA and LMP, which both have 3 gene deletions which prevent it from being infective. They also developed an adenovirus vaccine containing 3 proteins from the virus called 85YFV. And the effectiveness of these vaccines were tested in a simultaneous and a prime and boost model across 6 months. We analyzed cell trends across CD4, CD8, and memory B cell files and the antibodies to determine which vaccines were most effective.
So we'll start with the materials and methods. We had 5 cohorts measured over or tracked over a 6 months period of time. Our first cohort was a control group. They received a sulfate buffered saline, which is known as PBS, and the other 4 were our exposed groups. We could measure them over the 6 months period of time. From there, we ran different analysis. We ran a MANOVA, pairwise t-test, and a non-parametric test. The reason why we ran a MANOVA is because we wanted to see the comparison over multiple groups across different cell measurements. We ran it against CD8, CD4, and memory B cell populations against PBS.
And then over here we have the antibody levels, which are measured across the 6 months, the blue being the control. And we saw that for all of the antibodies, they retained elevated levels up to 6 months. And the conclusions of this project are that all of the vaccines were still viable, they're still durable up to 6 months, but there was no significant difference between any of the vaccines. When the mice were challenged, the 4 groups given the vaccine all survived, while the control group that didn't get the vaccine, all of the mice died in that group. Although all the vaccines perform similarly, it is to note that in areas where the plague is most relevant, the simultaneous model would be likely the most effective.
So before joining the CIS program with UTMB, I didn't really know what to expect in microbiology and immunology and biostatistics. This program helped me understand different areas in research in biostatistics. What I'll take away from this program is different level understanding of applying statistics to different areas in public health. And I'll be taking that interest to my grad school, or future grad school, which is USC, the University of Southern California.
Yeah, and for me, I'm a math major, so I didn't really know where I wanted to go with it, and the program here really solidified that I do want to go into biostatistics. I learned a lot of new statistical methods I'd never known before, and not only how to do them, but when it's appropriate to do them. And I've just really enjoyed learning about stats and biostatistics.